Let me give a quick revision of what we have seen in this slide. First, suppose this is a cell here and this is our desired gene present here. Let me give an example of insulin gene. Suppose this is a human insulin gene. I want this red color gene which codes for human insulin. What will I do? I will first isolation of DNA. What I will do first? Isolation of DNA. So now in this I am having this DNA, and here since this DNA is very long which contains the human insulin gene. Now what I have to do next to get my gene of interest? First step, I will do isolation of DNA. Second, what will I do? I will just cut this DNA into small fragments. We can call it fragmentation of DNA. How will I do it? Fragmentation of DNA by restriction endonuclease is an enzyme, right? I will cut this entire DNA. This entire DNA is now being cut into small fragments by an enzyme. What is that enzyme we call? We call it restriction enzyme. This is my gene of interest, right? Now third is, now I have to separate this gene from the rest of this gene, right? So separation of gene of interest. Separation of gene of interest. Now here see, this DNA contains lot of informations, lot of different genes, but I want only insulin gene. How will I do that? How will I extract the insulin gene? I will extract it with the help of a process we call it gel electrophoresis. What will I do? This is a gel, suppose these are wells. So I will place all the DNAs here. Yes, now DNA will start moving according to their molecular weight, right? Now if say this is my gene of interest, this one, this reached here. So now how will I know that this is our gene of interest? Because I must be knowing the base pairs. Here if it is 4 base pairs, 3 base pairs, here suppose this is 2 base pair. So this is being smaller in size, it will move faster, right? So we use gel electrophoresis. What is gel electrophoresis? If I tell about gel electrophoresis, it is the movement of DNA to oppositely charged anode in the presence of electric field. In the presence of electric field. This is what we called what? Electrophoresis. Now movement of DNA in the presence of electric field, DNA being negatively charged, it will move. Say here this is a cathode. So this is anode and this is cathode. So DNA will move to positive charge, and this movement is based on molecular size. Smaller is the fragment, faster is the movement, and our fragment is supposed to base pair. So here it is very small compared to other fragments, so it will move faster. So this is our DNA. Now I will this becomes transparent. Here I have shown you color, but actually it is very transparent. So what will I be using? I'll be using a ethidium bromide. What it will do? It will give a pink orange color. It will give orange color to the band, DNA band. When illuminated, when we put it in a UV transilluminator, this ethidium bromide will give a orange color band. Now we will extract this particular gel along with the DNA from this remaining gel, and this process we call it elution. Clear? After separation of DNA, what will I do? I'll do amplification. Amplification means I'll produce more copies of my gene. Amplification of gene of interest. Amplification of gene of interest. That means what will I be doing? From one I will produce many copies of my gene, right? Now after amplification, what will I do? I will try to ligate it with a ligation with a vector. Ligation with a vector. Ligation with a vector. How will I ligate it? Suppose this is my gene. What I will do, I'll take a bacterial cell. Say I have taken any bacterial cell, and this is bacterial chromosome, this is plasmid. So I took out the plasmid, I cut the plasmid. Now what will I do? I will just ligate this. I'll just put this here. And who will helping in this process? This process is done with the help of an enzyme known as DNA ligase. So after addition of DNA ligase, what will I get? I'll get my recombinant DNA. How it looks like? It looks like this. This is our recombinant DNA. Right? Now here I have inserted this with a vector. Now this is my recombinant DNA. Now what I have to do? I have to insert it. Yes, transfer it into the host cell. Now transfer into host cell. Now I will transfer this recombinant DNA into host cell. Right? So what is the recombinant? Let me draw the recombinant DNA here. This is my recombinant DNA. Right? Now what will I do? I'll put it inside the host cell. Now host cells can be plant, animal, bacterial cell. It can be bacterial cell, can be plant cell, and can be animal cell. If it is bacterial cell, what is the process we use? We use a process known as heat shock. What we do, this bacterial cell, if this is a bacterial cell, we'll treat this cell with calcium. Then we will put the cell in ice. Then we'll give 42 degrees centigrade temperature. Again we put it in the ice. Now after some time, after this, what will you see? You will see the recombinant DNA getting inside into the into the bacterial cell. Right? This is what. Now if it is a plant cell, what will I do? We'll be using gene gun. We'll be using gene gun. That means this DNA is coated in a gold or tungsten, and we'll put it in a, we just expose it to a high velocity, and then it comes in contact with the plant cell. Right? That is what we call gene gun. And for animal cell, what we use? For animal cell, we use what? Micro injection. Micro injection. Now DNA is inserted. If this is an animal cell here and this is the nucleus. Now here directly we will, this is already existing DNA. Now here we directly introduce the recombinant DNA. Right? We directly introduce this recombinant DNA. Right? This is what transfer into the host cells. Now once transfer is done into the host cells, what we do? We'll start growing the host cells into nutrient media. Culturing of the host cells into the nutrient media. Culturing of the host cell into nutrient media. Let me take the example of bacterial cell. If this is a bacterial cell, what will I do? I'll put this bacterial cell. If this is a bacterial cell, we'll put this in a fermenter. And in fermenter, two types of process will be there: batch, continuous. Any one I will be using. In case of batch, only one time nutrient is added. In case of batch, only one time nutrient is added. And in case of continuous, multiple times nutrients are added. Right? And after that, what will happen? Once the cells keeps on growing, after that, what will we do? We will do the downstream processing. We'll do what? Downstream processing. What is downstream processing? That is extraction of product. Downstream processing. What is that downstream processing? It is extraction. Downstream processing is what? Isolation of desired product, or extraction and isolation of desired product. Once we have the product, what we do after product? We will do separation and purification, separation and purification. For purification, you can use solvent extraction, paper chromatography, distillation. Then we'll add preservatives, then quality control, clinical trials, and then marketing. Done. Your product is ready now. Yes, with this we came to the end of the steps of recombinant DNA technology. Thank you so much for watching the session. Any doubt, please type in the comment section. Thank you.